So in my meeting, attending the meetings for EJPRD, it's very clear that most people are not in the metabolomics field yet, or connecting up to it. So I decided to give you a bit of an introduction. So what is a metabolite? It's a substance formed in or necessary for metabolism. The metabolome, you'll be comfortable with the terms genome and proteome. So there's also metabolome. And the field is called Metabolomics. So it's the scientific study of the set of metabolites present within an organism, cell, or tissue. And that on your right-hand side is Galen, way back a long time ago. They first invented it when they looked at your urine to try and figure out what it is that was wrong with you. So what are metabolites? They're a small molecule. They're physically and chemically very, very complex. There's a vast range of chemical structures and properties. Polarity, solubility, anything you can make as complicated as possible, that's metabolites. There's also a very wide concentration range, and its existence, any metabolite's existence, is very dynamic, with constant synthesis, absorption, degradation, and modification. So it's really very different to proteins. So we typically think of two kinds of metabolites: the primary ones, which are directly involved in normal growth, development, and reproduction. And then there's secondary metabolites, which are not required for primary processes, but have an important ecological role or other function. So antibiotics are an example of those, but also plants produce things to make sure predators don't eat them. They can grow without it. It's just an added protection. So what is metabolomics used for? So the field has been around for quite a long time in the agricultural area, because it's very tied into how you manipulate plants so that they can grow in the best conditions or have the right flavors or components in them. So for instance, there's an awful lot of work done at the John Innes Institute, not far from here in Cambridge, where they try to make sure that tomatoes have all of those wonderful things that are good for our health. The other thing that it's always been very strong for is for product and stress testing in food industries. It is making sure that that Parmesan that you think it is actually Parmesan and not something else being flogged. And also for the identification of harmful bacterial strains in your food. But the main development now, which is really transforming metabolomics and putting a lot of money into the field from all aspects, is for healthcare. And while you have your genetics and you have your proteins, metabolomics is really what's going to help push the personalized medicine that we've all been promised. But it's complicated. So while humans, roughly, we don't know, have about 60,000 metabolome, about only 3% of which are identified, they come from all different things. So your metabolome is not just what comes that you make, it's everything that you eat, everything that you're exposed to, your environmental, what you breathe in, what you are exposed to when you clean your house, all of that. So it is very much at the top pyramid of what a human or any organism is, because it is not just what you are, but also what you're influenced by. And that's why it's incredibly complementary to genomic and proteomic studies. So the metabolites basically represent the downstream biochemical products, which are much closer to the phenotype. You can predict something based on a gene or by the absence or presence of a protein. But just because the protein is there doesn't mean it's actually doing what it's supposed to. That's what the metabolites let you know. And that's why it's easier to correlate metabolomic profiles with phenotype compared to the other omics. But we need all of them. So our greatest chance in finding useful disease and stress biomarkers will be studies that show the correlation between all of them. And that's why we are hopefully contributing to EJPRD. Further complication. So I'm not saying genomics and proteomics is easy. I've worked in the field for a very long time. Before I came to Metabolites, I was the co-lead at the EBI for Uniprot. But genomics is 4 bases. Proteomics has 20 amino acids. And there's a lot of metabolites. It's a lot. The other thing is we know now, roughly, it says here 22,000 genes, but I think it's more 20,000 protein coding ones. And then 5,000 to 10,000 proteins you can identify with proteomics in a study. While in an average metabolomics study, if you identify 200 out of those 10 to the 5 plus chemicals, that's going well. Often it's much less than that. So as I mentioned, Galen was around a long time ago with his urine wheel. You looked at the color, you smelled it, you tasted it. It was difficult to be a doctor in those days. And that's how you diagnosed illness. Then in the 1940s, metabolomics started to take off. For those of you with a chemistry or biochemistry background, you'll remember paper chromatography, or maybe in your art class you did it as well. That began to be able to see what was going on. Then in the 70s and 80s, it began to actually have real applications. So for instance, diabetes. And then Oliver in 1998 coined the concept of metabolome. So it hasn't really been around that long. The most typical techniques that are used for this is mass spectrometry and NMR. Mostly it's mass spectrometry at the moment, but NMR is far more specific, and so when people are drilling down, they often use a combination of both. Another aspect that is very important for metabolomics at the moment is pushing into imaging. So with imaging, you can actually see where the metabolites are. And where this is useful is that you can actually look at a tumor without dissecting it, not hurting the patient, and see where the damage is. But also you can see where the drugs are actually reaching the tumor. Quite often they don't, they go off and do something else entirely. So I didn't have time to find a particular rare disease ones, but basically metabolomics is proving to be incredibly useful in oncology for stratifying the actual types of cancer that people have. It is very challenging sometimes to know at the beginning what form of the cancer and to predict or to recommend the best course of treatment. And also it enables you to track how far the cancer has gone based on the metabolites that are present. The other really good thing is that you can have very rapid and non-invasive diagnosis of the presence and severity of coronary heart disease. So you know whether to intervene or not, or appropriately would probably be better. And then also coming back again to plants, there's showing that each genotype has a distinct metabolic profile. And that enables you to know exactly what kind of plant you're dealing with. And then a very, very simple example for many of you who might know someone with diabetes. I have type 1 diabetes in my family, and them constantly having to prick their fingers, and now all they have to do is look at their iPhone and it's all sorted. Well, there's a little bit of an implant going on as well, but it does enable people to control their diabetes far better than in the past, because the highs and lows really do long-term damage, and it's best avoided at all costs. So coming back to where metabolomics fits in, so as I indicated, metabolomics is quite new to the party, effectively. So our focus has been in learning from what the other omics have done in the past, so that we learn and move on. So there are various aspects of what's challenging about metabolomics right now. First of all, it's across many species and many sample types and platforms. And as you can see from the growth chart below, the data has pretty much exploded in the last 3 years. I took over in January 2017, and the community began to submit. I don't think that was directly related to me, but it was a timely time to take over a resource which traditionally had been more of a research effort. But in 2017, well, late 2016, the directors of the EBI decided that Metabolites should become a proper service resource for our wider community. So what is Metabolites? It basically consists of 2 components: the repository for the studies and then the compound library. And the compound library is basically a library of all those metabolites that we have managed, or our submitters have managed to identify and are tagged with KEBI identifiers for easy use by the other omics. So as I said, the study repository, it contains detailed study protocols, raw data. We're the only resource at the moment that insists on the raw data. And that's because that enables people to come back and reanalyze as the field goes forward. Otherwise, you just get an Excel spreadsheet of what people had identified at that particular snapshot in time. And while it was very good at that point, science does move on, thankfully, and you can go back and look at it again. It also contains annotated metabolites, and we try to connect it to biological pathways. In the last couple of years, we put in a new website and a granular backend, a new submission process, and it is an Elixir recommended repository, which is why we ended up in EJP. Then in the compounds reference library, you have the actual chemistry, and then the connection to biology again, and through to pathways and reactions. So RIA, reactome, wiki pathways are all component from here. In the past, it was purely the compounds or the metabolites that were identified in the studies that were submitted to us. But there's a great need for a centralized library. Identifying a metabolite is actually very, very expensive because you have to compare it to references, and you buy those, and they are expensive out there in the world. And so many people do not want, first of all, they don't have access to a wide range. And second of all, they don't really want to share because they've already paid a lot of money for it. So it's only by pulling things together and putting them in a compound library like this that lots of people can actually benefit who don't have the money to have references of their own. So we are trying to become a centralized library, and part of that is also a sustainability thing. So for instance, the Irish Marine Biodiscovery Database, which tracks metabolites in the coastal regions of Ireland, submitted some of the metabolites they had identified, and we did that work in collaboration with KEBI. So in the context of clinical data, this is a big change for metabolites in the last few years, and we do follow Metabolomics Standards Initiative. There have been templates developed for the deposition of more specific data. This was fine at a certain point, but it is needing to evolve as more and more clinical trials are being done, including metabolomics data. And we are trying to learn from our colleagues in the other omics and through this project of EJPRD to actually deliver on this. As you'll see from this little slide, Homo sapiens and mus musculus are now more than half the database. The challenge is that many of those metabolites are not actually known that are submitted to us. So of all the metabolites we have in the database, 70% are unknown. So we capture as much data as we can and hope that somebody can come back at a later date and re-identify them. Again, that's why it's really so important to have the raw data. We did not reinvent the wheel. One of the first things we did in 2017 was to map as much as possible of the data that we captured to ontologies. Our previous work with Kebi and many others really facilitated this. And so we submit new terms to the appropriate ontologies so that we can use them. We do have a small internal control vocabulary for instrumentation because there hasn't traditionally been an ontology for that. But the University of Oxford is now developing one, and they have a lot of experience with ontologies, as many of you will be aware. And so we will then, we have shared already our ontology information. There's also an effort in Germany in this way. And again, we're collaborating to ensure we all share it, and share the work and share the data. So as I mentioned about the study submission, we redeveloped it, but it's important to realize that we have very different approaches to this. So you have the individual researcher or a lab who sends the data, and he goes through an online guided submission process, which every step of the way they're told what is needed and it's validated on that page before they're allowed to get to the next stage. We have large phenome centres submitting data to us in the UK at Imperial and Birmingham, and shortly Liverpool. And we also have a collaboration with industry. So Metabolon offers submission to us as part of their service. So they deposit the raw data with us, and then the study is handed over to the researcher with the raw data and identifications. And then the researcher takes it over. This has also very much driven how much human data that we're getting, because Metabolon is used quite extensively in clinical research. As I mentioned, we try really hard to work on the ontologies, and we help our users by doing that, by giving them these prompts so that there is a minimum possibility for getting it wrong. Of course, if you are very adventurous, like some of our users are, they still seem to manage it, but we do our best to help them every step of the way. And again, the amount of data is so huge, we try to make it as easy as possible for people to manipulate whole chunks of data. This might not look really interesting for many of you, but our users absolutely love it. The other thing is, for those who come and read the studies afterwards on the website, we enabled that the protocols are visually linked to the assays, because you can have many different assays in a study depending on what they're trying to do, and knowing how they actually did the protocol is really important for reproducibility. So another key point, which is probably interesting for EJPRD, is that we get data from all over the world. While we are at the European Bioinformatics Institute, our biggest emitters are actually China, and they submit an awful lot of human data. Our second submitter, biggest country, is USA, followed by the UK, Germany, France, and so on and so forth. And that means that we do pull in a lot of data from all over the world, which should hopefully help with raw rare diseases data. You can also see there what I mentioned about Homo sapiens, how the amount that is submitted per year has massively increased. And also how much we process each year. And then I put in the bit about the diagram of the journals, because in the past we were primarily in very specific journals like the Journal of Analytical Chemistry or Metabolomics, but now more than half is the Nature group or PLOS, which really reflects the fact that metabolomics has moved firmly into the clinical area, human area. And so this is, again, to stress that our users are really, our submitters are really across the world and doing very many different types of things. But a lot of it is about taking care of people and trying to improve health. So our goals are to add value to the studies with additional expert curation. We also want to add confidence and evidence attribution. We just got a grant from the BBSRC and NSF in America to do this starting in January, so that'll be really exciting. It will also enable us to extend the compound reference library into a comprehensive fair data set for identified metabolites. And the other key thing is to enable reanalysis of studies by other researchers as the science progresses. As I mentioned earlier, having that raw data enables us to do that, but we need to make sure that that the evidence or the trail of how they actually predict the new things is captured so that people can have confidence in it. So we're involved in very many data standards efforts to ensure that we try and standardize the data, but because we are also part of the omics world at EBI, it enables us to make sure those standards work across the omics. We're also part of the metabolite identification process, so we have a number of grants with researchers in the labs and tool developers so that we can contribute to ensuring that the metabolites are identified as confidently as possible, and that's captured in both the studies at the in metabolites, but also enabling us to provide training to people who come to EBI, which a lot do, and on our online offering, so that people who are coming from backgrounds that are not used to metabolomics, which is actually quite a lot, the community is incredibly evolving at the moment. It's quite a challenge to enable people to understand what metabolomics is when often they don't even have a chemistry background, but that's part of our role. We're in a whole ecosystem of metabolomics resources which have grown up over time, and we do our best to collaborate in this ecosystem. So some aspects are working with our American colleagues, Metabolomics Workbench and Metabolonote, that's what it was supposed to be called, it's now Metabobank in Japan. We're having bi-weekly meetings to help them get up and running. They will copy the metabolites structure, and we intend to exchange data. So going forward, we will not only have the data that's submitted directly to us from all over the world, we will also have an exchange with our American and Japanese partners, so that if you come to any one of our sites, you will have the full public metabolomics studies. We've already worked on Metabolomics Workbench with lipid maps. Lipids are, while metabolomics is challenging, lipidomics is even more challenging, and it's really important that we work together. As many of you will know, lipids are often incredibly important in disease. We are also working with GMPS Massive, which is another natural products resource, which is also really helpful for enabling us to discover the human metabolome, because we'll then know when we see something, is it something that the patient has absorbed from the environment, or is it something they ate? I do remember being at a Metabolomics conference where they had spent an awful lot of time and money trying to track down what they thought was a disease biomarker, and it turned out to be a component of the lady's perfume. They were not impressed, but that's science for you. It's already possible to link chromosomic data submissions through the MetaboLights. You'll be aware of EGA, ENA, Pride, and Array Express. And now we are connecting Metabolomics into this. We are also trying to develop a controlled access capabilities along with Pride and EGA, because there are more and more multi-omics studies. I had an email exchange with Peter Brown that many of you will be aware of earlier today about pushing on one of the studies he has. We haven't yet had any studies from EJP, and that would be something I'd like to understand, but we are getting studies from elsewhere. And then one of the nice things about working at the EBI is that you can connect through the different omics techniques and resources. So we're trying really hard now to integrate Metabolomics into things like the enzyme portal, so that you can go from identification of a metabolite through PDB, Uniprot, Ensemble, or any flavor you choose. So we're always welcoming feedback. Is there any features you would like to see included? What other kind of data would you like to capture? And what interactions with other resources would you like to see? And also, as I mentioned, we haven't yet had any use cases for controlled access. From the EJPRD, I am aware that in certain countries, this data is more challenging to share than others. But as you will be aware, the EGA is moving to a federated model, and I expect the same will happen to metabolites at some point once we develop the controlled access variation. And then finally, but not least, all this work is done by a very small but very friendly team. So do reach out to us if you have any questions at all. We'd be delighted to hear from you. And thank you for your attention.